topic of discussion is theory of slide making. So, take this is a clean slide. So, in this clean slide, first we are applying diluted cell suspension, then after that we are applying fixative, then we are applying the heat by keeping the slide on a slide heating chamber. So, now let us understand the theory behind the to get a good metaphase spread. Take this is the side view where the single line represents the fixative layer, the double line represents the glass slide surface, and this is the swelling cell. The cell has become swelled due to treatment of hypotonic treatment, and these are the chromosomes. Now, when we apply the fixative, initially the swelling cell is supported by the many layers of fixative, but with the time passes due to heat, the fixative start evaporating, so that the fixative layer becomes thinner with time. Now again, as the time goes on, the layer of fixative become more thinner and thinner, and the meniscus of the fixative pushed down on the top of the cell, so that the cell is now pressed in between the meniscus of the fixative and surface of the glass slide, and finally the cell ruptured and a well spread metaphase has come out. So, this is happening with the time. First, the cell is supported by the many layer of fixative, and as the time passes, the fixative start evaporating, and finally the layer of the fixative become thinner, and the meniscus of the fixative pushed down on the top of the cell, so that the cell is now pressed in between the fixative and the glass slide, and finally the cell is ruptured and a well spread metaphase has come out. This is the metaphase spread. So, here to get a good metaphase spread, the timing is very important. For example, if we suddenly give a very high amount of heat, so what will happen? The fixative will evaporate suddenly, and the cell will rupture immediately, so that the cell did not get sufficient time for enlargement, due to which the chromosomes those are present inside the cell, they did not get sufficient space for their position, and when the cell ruptured, a clumpy metaphase spread we will get. So, sudden heat we will get a clumpy metaphase. If the timing is very slow and the fixative taking very long time to evaporate, then what will happen? There will be a hole formation, starting hole formation on the cell membrane, due to which there is chance that some of the chromosome they may come out of the hole through this hole. So that what will happen, the metaphase spread what we will get after rupturing of the cell, it will be not complete, there will be loss of chromosomes. So if fixative taking very long time, then there will be loss of chromosomes, and if fixative is evaporating suddenly, then there will be a clump of chromosomes. So we have to standardize the evaporation time, and there is no any fixed timing that how much time we have to give. It depends, it need to be standardized from lab to lab. So this is the mechanism behind to get a good metaphase spread. Thank you. This is the image of a well spread metaphase. Here we are seeing how metaphases are well separated from each other. So here we can easily identify the chromosomes, and it is easy to analyze the chromosome. This is the image of a clumpy metaphase. Here we are seeing chromosomes are overlapped each other, so it is very difficult to identify, and it is very difficult to analyze them also. This metaphase showing there are loss of chromosomes.